over there.
Yeah, I've been to the hospital, which is, the
military hospital, and there are two. One is SLM, is a civil hospital, and another is a military hospital, GH 153.
I think one of those.
Is this working?
We had a pulmonary hypertension conference.
It must be at GH 153. Dr. Moore.
Yeah, we can hear now. Yes,
Okay, thank you, thank you. So I think what is the next session? Professor. Professor Dash, right? I think that's how the schedule is. So Dr. D Dash is presenting in person here. So I'll give him the mic. Just try to.
In the program, Shweta Saxena will be speaking, right?
Yeah, there has been a bit of a reschedule because Dr. Dash has to go back and fly back. So we requested if that can be accommodated first. So we are giving that to Dr. Dash. Shweta, is that okay with you, right? So we'll just.
Very much okay.
Yeah, okay, thank you, Dr. Shweta. Thanks.
This is working.
That is working.
That's all right. Hello.
It's working.
It's working. You can use that.
Okay, so
Working.
And there's no sound.
So after all these cyber activities and online speakers, now I am here in person, very much physically. But my speech, my talk will be in line with my esteemed earlier speakers, Dr. Mishra, Dr. Sharma, etc. So it will be in sync with this. But while we talk about the heart problems, there is a very mischievous player there, and that is known as platelets, very small blood cell. That creates a lot of problems. It's a very active cell, very active. So now I shall start with, thanks, madam. So I shall start with my mentor, whom I joined at BHU in 1982, and I did my MD till 85 on the ribosomes, bacterial ribosomes. Now, after I became a faculty immediate next year, I thought that I should continue on the ribosome, but I thought that it's a very difficult task because it requires water centrifugation preparation. So I chose a cell system that should be very easy to pick up. Blood cell. Blood is very easy to handle. And in the blood there are RBCs, neutrophils, platelets. RBCs many people are working. Neutrophil at that time, Professor Talwar, he instituted National Institute of Immunology. I thought that I cannot compare with a figure like Talwar. So I thought that the easiest thing to work on platelets. But I did not know it would become a nightmare for me. It is a nightmare for a person in which it becomes active. And it is very difficult for me also to handle platelets. The moment you prepare platelets, it becomes active in the test tube. So it is so active, as active as nerve cells. Okay. So in the next slide, I shall try to show you a video because it actually works.
I'll do that.
Rahul can do it.
Yeah, I can do it, sir. You just tell me next slide and do that.
Now this video. Okay, video is not working. Okay, so that video I have picked up from the YouTube. It was showing that how dangerous platelets can be. Platelets are our friends. They are not enemies. Because whenever you have caught any injury, without our knowledge, that healed up by action of platelets, known as hemostasis. But as you age, come to my age, you are afraid of heart problems. You are not afraid of any injury. And all the time, we have to keep our platelets very happy, calm and quiet. And if there is a cardiac problem, the drugs they give, first line of drugs, antiplatelet drugs. Okay? Dr. Mishra also mentioned something. Now, here, you see these are very resting, very happy platelets, as you see under a microscope. Once they get active, you can see their morphology phenotype. There is micro phallopodia, lamellipodia, and microspikes, and all those things. When they become active, and very easily they can become active, they spread out. They spread their morphology, secret many things. My basis of my talk would be, we are all biochemists. I see very few biochemists here in the audience, mostly students. But whatever the come in the mind, basically how platelets should behave, as you see, they contradict all the opinions. How platelets goes against our view, basic biochemical views. That will be my talk, and it mostly based on my lab, the results from my own lab, mostly majority, which have been already published. So now coming to that, platelets can change their morphology. You understand that it requires a lot of energy. So we just asked some time back, I think 5 years back, what is the source of energy? How they get so much energy? And once you show energy, because we are biochemists, we shall think of the glucose metabolism. So okay, so we know the answer, let us try. So if you see you are in the first year, if you are being studied glycolysis, then you can see that these are the energy yielding tags that platelet do. They do a lot of things. If you have a clot, after 1 hour, you see the clot will become 1/3 of its volume. Clot will retract like a muscle man pulling the clot. So this muscular job also is done by platelets. If you inhibit the platelets, clot will not retract. So platelets require a tremendous amount of energy. We wanted to see how they get this energy. So we just studied a little bit of metabolism. We recapitulated our undergraduate information, and you can see that here it is a glucose. It is by glycolysis converted to pyruvate, if you know this. Pyruvate has 2 phases. It can go into TCA cycle like this. If you block the TCA cycle in the fermentation, it can go to become lactate. When the mitochondrial respiration, we know all this. What we want to tell that during this glycolysis, it does not require oxygen. Entirely anaerobic, no oxygen, no molecular oxygen required. In contrast, if we go for TCA cycle, now here it is not showing here, the pyruvate is there, it enters the TCA cycle, and TCA cycle requires oxygen. That oxygen we inhale, we take oxygen. That oxygen going into the blood through hemoglobin, then enters the cell, enters the mitochondria. And inside the mitochondria, TCA cycle that game takes place, it releases a lot of ATP. And that should be the secret of power of platelets. We thought, we did an experiment in our lab, and in this experiment, this is an oxygraph experiment. It shows the mitochondrial oxygen consumption rate. You can, this red line shows that how much oxygen is consumed. Normally, happy mitochondria is routine. We added a stimulant like thrombin. We expected a peak. It will consume oxygen because TCA cycle will take place. It did that. There is a peak. But lo and behold, just after a moment, after a few seconds, oxygen consumption dropped. We have not expected. We thought that oxygen will remain, consumption will remain high. Then, without oxygen, less energy, how platelet should do the job? Now, there is a drop in oxygen consumption. Now, we just contemporaneously, in parallel, we compare with the aggregation. This is the platelet which is getting aggregated. In parallel, simultaneously, if we study the oxygen consumption, you see that platelet aggregation is exponential phase. It is getting too much aggregated. But you see oxygen consumption has already dropped, wherefrom it is getting energy. Then we thought of a gentleman in 1952. He discovered, his name was Otto Warburg, and he discovered an effect for cancer cells. That is known as Warburg effect. For cancer cells, not for any cells. That was their cancer cells, they gain their they metabolize glucose by only glycolysis, no mitochondrial respiration. So abrupt fall in oxygen consumption coincided with rising phase of aggregation. So exclamatory mark, how it happens. Why does mitochondrial respiration drop in stimulated platelets, stimulation, quote unquote? So we thought of, is there a aerobic glycolysis Warburg effect? We have never thought of. In the platelets, cell like platelets, cancer cells, they grow. For the growth, they require a lot of biomass, lot of material. Therefore, they don't bond their glucose, bond is to Hawa, just glucose to carbon dioxide and energy. They don't do that because they keep that material for new cell division. Platelets are not growing. Dead end cell, 10 days lifespan. So while platelets will undergo Warburg effect, that was the question we asked. Then I shall not show the experiment. In this cartoon, I shall show that what happens in the Warburg effect. Here you can see that in Warburg effect, only energy, all the energy will be generated from glycolysis, no TCA cycle. So lot of glucose, it requires a lot of glucose. So this is a glucose transporter. In a large amount, it translocated to the membrane. Lot of glucose will be input. In normal cells, so much glucose is not required. Here, lot of glucose is input. Glycolysis takes place, but there won't be any mitochondrial respiration. I have put a cross here because no mitochondrial respiration. It will go to the doctor. So the hallmark of Warburg effect is experimentally more of glucose receptor on the membrane, more of glucose influx, more of lactate release. Three hallmarks. We did in our experiment, we stood by that and it proved, we confirmed that. I am not going to show the result, and we confirm that aerobic glycolysis Warburg effect takes place in platelets, stimulated platelets. If you see the energetics, glycolysis full TCA cycle is 27 ATP, that is expected lot of ATP. Only glycolysis, you see only 2 ATP. 27, 13, 15 times less. So more of glycolysis should take place to compensate, and platelets requires a lot of energy. Why platelet will be doing this? This is the question you ask, how and why. That was the question I asked. We asked, I think, in the 2018 or 17, the students, they did a lot of labor at the time, and we justified that. Aerobic glycolysis justified in cancer cells, but not in platelet. We did a more experiment, and I shall show the final result here. That is a mystery. So I saw that real biochemistry is not followed. My title of matter, Adventurism in biochemistry. If you just do a lot of adventure in the area of biochemistry. So here you can see that more of group receptor group 3 in the platelets, more of group, more of glycolysis is there, what takes place. But why it is required for the platelets? What happens once there is a lot of this is blocked, mitochondrial respiration blocked, this is pooled, these intermediates are pooled amount, so pentose phosphate pathway, the hexose monophosphate pathway will be stimulated. So through hexose monophosphate pathway, lot of NADPH is generated, which we know in the adipocytes, lot of NADPH is generated. Now there is an enzyme is NADPH oxidase, NOx, NADPH oxidase. So NADPH will be acted upon by NOx, lot of reactive oxygen species is produced. Platelets do all the diverted their metabolic process only to get lot of loss. Loss is not bad for platelet, it is good for platelets. It requires a lot of reactive oxygen species. You will ask me why, how it is good. Look at these integrins. These are the fibrinogen receptor or aggregation takes place through this. Just you have a look at this through the rods, you can see. So you can see, again I can show you, now this is a conformational change in presence of ROS. Now it is ready to bind fibrinogen. Platelets are ready to aggregate. That is the goal of platelet. Platelets are 10 days lifespan, and only goal of platelets to get active. That is the essence of platelets life. So to do this, it requires ROS. So this paper was published in Hematologica long back in 2019. Mystery does not stop here. For platelet is such a cell There is numerous mysteries. There are many labs, and there is a gentleman, Andrew Wells, and he has done a lot of experiments to show that how platelets are very unusual. Platelets don't have nucleus, no genomic DNA, no nucleus, no DNA, nothing, but they synthesize proteins. No messenger RNA, so they don't have gene. They get messenger RNA from precursor cells, from the megakaryocytes. They inherit those messenger RNA and they are functional. They are not just inert that they have got from their ancestors. They are functional. And they process the protein synthesis, including ribosome. They have ribosomes also. And they have also microRNA to regulate. So it is a full-fledged machine. They are destined to synthesize protein, but they don't have DNA. It was discovered by Andrew Wells. And it is a very good work. And they have, in the sequence of papers, they have shown that placogen activator, inhibitor 1, tissue factor, BC. So a lot of proteins are getting synthesized customizedly in platelets. But what was nature regulation still in darkness. We also found after that, because just Dr. Saxena madam was talking about hypoxia, we have shown that under hypoxia, platelets, they synthesize hypoxia-induced factor 2 from pre-existing messenger RNA. Platelets have messenger RNA transcript for that, they synthesize also, and that also we published same year in 2019 in the same journal, Hematologica, where it was shown that relief from vascular occlusion, it is title is different, I haven't seen the title, another paper title. It is not hypoxia in the Hematologica, I have Now coming back, there is another surprising scope in the practice. That is our current research area I am going to share with you. There are pathways like Quint pathway. Sonic Hedgehog pathways are funny names. In the science there are funny names. Like you see, there is a protein known as sauce. Name of the sauce is son of sevenless. Then they discover another protein, they give a name, daughter of sevenless. So here, Sonic Hedgehog pathway is a signaling pathways, another is Notch pathways. So these pathways they operate in embryonic cells and cancer. Because by this cell grow, cell differentiate. The embryonic cell, they will stem cells, they will differentiate. So these are very much dedicated to that cell system, not to platelets, which is a dead and well differentiated cell. Platelets don't grow. Platelets don't produce cells, next generation cells. So they should not be there in the platelets, but we found that this pathway, Sonic Hedgehog pathway, is present in the platelets. Absolutely unbelievable. And with Mr. Hedgehog, I didn't know. I saw on the net, I saw there is a movie, video game, in 5 years back, I think. Sonic Hedgehog, yes, Sonic the Hedgehog. Then I went for an examination or something to Rajasthan Central University or selection committee somewhere. And the person who has invited me, he called me a dinner to his home. And he told that, Dr. Das, keep your shoe in a safe place. I told, why? Now, hedgehog may enter. मैं बोला कि hedgehog यहीं पर है, मैं खोज रहा हूँ hedgehog क्या? Then I saw the net, and this is the look of harmless, spiny, small, and good creatures. And in the Rajasthan Central University campus, you go, you put your shoe, I think you will find some hedgehog within your shoe. So therefore, now, then I thought about little bit, want to know more about hedgehog in the net. Once I Googled, I found that Sonic Hedgehog in theaters 2020. Corona, everything was closed. I have not gone that movie. Then again I saw that movie must have been very hit. Sonic Hedgehog in theaters in April 2022. So I am not a very much cartoon fan, my cartoon is up to only Tom and Jerry. So, but the students, if you go and see Netflix or somewhere, if you see that these movies are available and they are very entertaining, please give me a message. I want to see the movie because now I have published a paper on this also. Now, this is a sonic hedgehog pathway which is operable in stem cells, embryonic stem cells. Now, these are the sonic hedgehog receptor. This is a cell. Sonic receptors are there. Now, here hedgehog is binding. Once the hedgehog is binding, there is a signaling. After the signaling, the final it reaches the nucleus in the DNA, genomic DNA, and there is a gene expression. Mind you, platelets don't have DNA. They don't have nucleus at all. But they have these proteins. Mind? Okay? So by Western we found that these smoothing receptor, patch receptor and the gly proteins all are present in the platelets. What is it going to do? That was the big question mark. So we did a lot of experiments. We found that by RT-PCR, we found that this messenger are the transcript of sonic hedgehog is present in the platelet. They are synthesized, sensitive to pyromycin. In the flow cytometry experiment, if you stimulate the platelets, the protein is synthesized. Not only synthesized, it is translocated, expressed on the membrane. It is reaching the membrane. So, ये भी होता है. You see that in the statistical significance, what it is doing, it has a platelet actin cytoskeleton, it is influencing. I am not going into detail at all. It will be too heavy before the launch. And then it contributes to platelet adhesion. Platelets get adhered on the immobilized collagen or fibrinogen. Now, if you inhibit the sonic hedgehog pathway by these inhibitors, bismodazine and cyclopamine, they are used now as a cancer cell, anti-cancer drug, because bismodazine already approved by the authority as an anti-cancer drug, and cyclopamine is not yet approved, but these are the very potential inhibitors of pathway of hedgehog signaling. But in platelets, if you give this, they are inhibiting the platelet activity. This is the platelet aggregation you can see. So you can see that this is a normal aggregation. If you give inhibitors, bismodazine, cyclopamine, aggregation significantly, it's inhibited. You see there is no aggregation here at all with the graph. Here with collagen, it is inhibited. With ADP also, it is inhibited. In mouse platelets also, we have done that thing. So it is inhibiting the inhibitor of sonic hedgehog, inhibiting the aggregation as well. Then we did a study in the dynamic adhesion. This is a type of experiment you may not know. It simulates the normal arterial shear 1500 in per second. Here you see that this is a normal adhesion of the platelets in presence of anti-sonic hedgehog drugs. It is inhibited, dramatically inhibited. Platelets are not allowed to get activated if you give sonic hedgehog inhibitor. So they have a very tremendous impact in platelet biology, which we don't know yet. So this is also we did the experiment that we in the live animal, it is known as live cell imaging, intravital microscopy. In the mesenteric artery of the mouse, we opened up the mesenteric artery, we made the platelets fluorescent, we let it aggregated here, and you see that there is a thrombosis found at 25 minutes. In presence of inhibitors, the extent of thrombus within the artery, mesenteric artery decrease. So that is a in vivo simulation is also there. So this is our cartoon final picture. This is what is we know here. Things are there. That what are the antiplatelet drugs we use? One drug we use aspirin. Aspirin inhibits thromboxane generation by inhibiting cyclooxygenase enzyme. Second drug, PY12 receptor inhibitor, ADP receptor inhibitor, for the clopidogrel and ticagrelor group of drugs. Now, ADP is released from one platelet and by paracrine effect, it activates the another inactive platelet. It is a paracrine effect. Clopidogrel will inhibit this receptor. We add to this thing that from platelets, sonic hedgehog can be released through exosomes. We also did the microparticle exosome. It is getting released. That we have shown that in that paper. It is released. So it can stimulate the neighboring on-active platelet. So we propose to our sonic hedgehog inhibitor like bismodazib, which is already FDA approved, and cyclopamine to the group of drugs. So this paper we have just this month it has come out, and the journal has put our work in the cover page. It has because so it is a very proud moment for our laboratory, my students and myself also. This is a broad advances journal, and it has put our figure 5P in the cover page. And this is now September issue, 30 September. So it is the issue which is now. It is a free online. You just Google broad advances. This issue you can find our paper. All these figures that I कहानी में जो बोला आप लोगों को मिल जाएगा. Now coming to very quickly another pathway NOTS. This work is ready. It is not yet published by us. So NOTS signaling also we have done. NOTS works by zostaglin because this is the NOTS receptor, NOTS ligand, and here is a NOTS receptor. This is one cell. This is another cell. When they come close together, then this ligand will attach physically with the receptor. Once this is known as zostaglin signaling, not paracrine zostaglin. Once they will have this signaling, there will be an enzyme which is known as gamma secretase, and gamma secretase will clip this NOTS receptor. This is the clipped fragment. It will again go to, it is not shown, again go to nucleus and express the gene. Platelets, again I reiterate, platelets don't have DNA, don't have nucleus. But still we showed that this is the artificial human platelets abundantly, not only expressed, abundantly expressed. The CQ value is quite low. Abundantly expressed NOTS1. They also abundantly express the ligand, DLL4, delta-like ligand 4. So ligand, receptor, all are expressed on platelets. And when the platelet aggregate, they come close together. The scenario is ready. They can come together physiologically, and which is not known. This ligand receptor can approximate very closely. So we did very quick experiments. We inhibit the gamma secretase. Gamma secretase inhibitors are commercially available again to inhibit NOTS signaling pathway in cancer context, anti-cancer. And they also inhibit amyloid beta production also, mind you. Same gamma secretase, same. Now all these experiments, and this is a platelet and neutrophil aggregate formation, platelet monocyte aggregate formation, and platelet aggregation, all are inhibited by DAB, which is a commercially available gamma secretase inhibitor. Again, we did the thrombus formation experiment. We did that. It is the control thrombosis found in 15-20 minutes in presence of DAB, this gamma secretase inhibitor. There is a blunting in the thrombus formation. This is the thromboelastogram experiment. It shows that thromboelastogram. It shows that okay. It also shows that thrombus formation is inhibited. Again, we have a cartoon for this. It should attract your attention. If you do, it may go to Google, but if you tell only science, it could capture your mind, attention. So for that, we made a cartoon. My student is very expert in all these jobs. Also, is good at science also. So here you see that this is our hypothesis. Platelets, they express DLL4, they express NOTS on their surface, so they can approximate. We proved it by giving an anti-DLL4 antibody. If you give incubate platelets with anti-DLL4 antibody, it will attach to DLL4 and block its introduction with NOTS1. So it is acts as a therapeutic antibody, Arciximab, RioPro also. So we did that experiment also and shown that this is a combination signal. Now, Dr. Where is also another funny thing. Our story from our lab is this much. 2 more slides. Where is has proposed a very funny thing. It is in the paper Blood in 2010. Platelets बच्चा पैदा कर सकता है. They don't have nucleus. They cannot divide, cell division does not take place. But the title you see that, it will catch attention. Anucleate platelets generate protein. Platelets are very unusual. Lot of mystery still lurking behind the platelet corridor. Just one has to look at the mystery and discover them. Because as some speaker told, Bill Clinton had a cardiac bypass surgery. Again he has a problem. That is what he told. A person, ex-president of America, cardiac bypass, he was treated. Again he has angina symptoms. So lot of mystery is still there. Discoveries are yet to be made. We cannot be complacent that we know it. No. So, and for this, when his picture shows that, you see that this is a megakaryocyte, it generates a platelet, proplatelet, the platelet board or proplatelet. This is the platelet, and it can, under microscope, it found that it can divide into two in microscopic. So, I think that ends my talk. My project, you see, it requires a lot of money, and so over a period of time, DST, DBT, and ICMR, our lab has been upgraded by ICMR to a center of advanced research 2 years back. So some good funding. So ICMR funding is there, and Tata Innovation Fellowship, and now I am getting the Sipos National Fellowship. So all these are the source of my funding for the lab. And this is a bit dated picture, old picture of my lab, because some are left. This gentleman, you can, this smart looking person, you can.
Very smart.
I think that
Still is very smart.
still, okay, that means I am not that smart. Okay. So, he is Mohammed Akhlaq, he is doing a PhD, brilliant, and this paper also he has a name, very good worker. And Sushil, he has already completed his PhD, now working as a research associate under me, scientist C under me. And Abhishek, he has joined MD under me, then he left, and now he joined MD in medicine. But he is very much obedient to me. This is a guest to my lab from UK. I forgot his name. And Deepa, she has done PhD, now she is in USA. Forrest, he has done MD as well as PhD under me. Now he is gone for a postdoc in USA for 1 year. Josna, she has also done MD and PhD under me. Now she is associate professor. And Arunati, she has completed PhD under me. Now she has a proud mother. Now she has come to Banaras. Her father is in Banaras. And Rashmi, she is also doing his PhD under me. So, thank you.
Thank you very much. Dash has given you a very good new information from the platelets, and I am very happy that you have presented this paper. If there are any questions, somebody would like to ask?
How proteins are synthesized when it is, how messenger RNA is synthesized there?
Platelets, they carry messenger RNA.
They are not having any nucleus.
No nucleus, yes. That is another mystery surrounding platelets. They carry this messenger RNA while they are generated from megakaryocyte. They carry, but it is not in this speed. It's supposed to be the messenger they carry the whole transcriptome that is carried from the megakaryocyte has a protein. But what is the purpose? Gradually these proteins are getting discovered. We have found Shunikheda, NOTS-related proteins, and hypoxia induction factor. These two we have found. And already they are all been discovered. So, a lot of things. If you know everything about platelets, you don't have heart attack.
How do they divide? How do they divide once they don't have got a nucleus? How do they produce progeny?
That is not our work. That is Dr. Wellich he has done. And I think he has
What is the mystery behind it?
That's the mystery.
that is something we've done it. But he has found a microscopic picture is there that evidence in certain conditions, in certain experimental conditions, he has found it. It is not that the platelets you are counting, then you are like, okay, मेरा पास 50,000 का मुझे 75 हो गया. बच्चा पैदा कर लिया, इसके बाद नाना नानी बन गया प्लेटलेट्स। ऐसा नहीं होगा कि आप देख रहे हैं वो बढ़ता जा रहा है, वो अंडर स्पेसिफिक कंडीशंस वो बढ़ रहा है, वो डिवाइड कर रहा है। So स्टूडेंट्स को ये करना है कि एक्सप्लोर करना है अनएक्सप्लोर्ड क्वेश्चन, ask questions which is not known, ask always inquisitive, ask questions and try to. Thank you. Sorry, you have some questions. Yeah, Dr. Brown, you want to ask something? Dr. Brown? I think we have been okay.
You can't do it,
So we can't hear.
we are already behind time, you know. So, I will request Dr. Dash for your very dashing lecture. Kindly accept this.
Dr. Dash himself is very nice.
Then I think we switch on to another lecture by Dr. Shweta.